So this is the second part of sensory physiology. Here we're going to handle what's going on in the ears. Ears, we get equilibrium or rotational movement, and then we get hearing. We get auditory sensation. And then finally, the last part, we'll finish up with the eyes, the most complex of the special senses. All right. So let's first look over the overall structure of what we call the inner ear, where our sensory organs are going to be housed for equilibrium and for hearing. And so if you remember from your anatomy days, we have this bony labyrinth located in that temporal portion of the brain or of the skull. All right, we have this bony labyrinth. It's basically like tunneling in the bone. Okay, the bony labyrinth rings through and it's filled. It has interstitial fluid, perilymph in it. It's very similar to what lies in the interstitial fluid, right? So it's high in sodium, low in potassium. Okay, but inside, running the length with it, is this inner membranous labyrinth, right? And this is where the sensory organs are going to be held for what we call the vestibular apparatus, this portion of the ear here, where we're going to get equilibrium, and then this portion also, the membranous labyrinth, is housed with endolymph. All right, and as the cochlea, this is where we're going to get auditory sensation, but those sensory organs are housed in the endolymph, and what we find in the endolymph is high in potassium. So we have a cell and it's bathed in endolymph. So endolymph is high in potassium. So it would be higher in potassium than what's in the cell. And so if there's a channel for potassium in this case, what's going to happen? Our potassium is going to rush in. And therefore, in this case, since we're housed in this endolymph, opening of potassium channels, instead of hyperpolarizing the cell, bringing the membrane potential down, now potassium's rushing in, and we're going to get depolarization. So just keep that in mind. It's a little bit backwards because we're housed in that envelope with the high potassium. All right, so we're going to look at equilibrium, which is housed in the vestibular apparatus, the back portion, all the portion besides the cochlea. There are two main structures. We have the otolith organs that contain the utrica and saccula. All right, and then we have the back portion where you saw those kind of rings. Those are our semicircular canals, and there'll be 3 semicircular canals that make up all the semicircular canals. All right, what we're going to see, so we'll look first at the utrica and saccula, and what this gives us is linear acceleration in one plane. Okay, think horizontal, front backwards, and up down. All right, so when you jump rope, what's your head doing? It's going up, down, up, down. Okay, that is your saccula. The sensory organs of the saccula are giving us our vertical acceleration, as if we're jump roping. Anybody recognize this? This is Montezuma's Revenge at knots, and it takes off very quickly in a horizontal direction. Okay. That is going to, organs that are going to be able to account for that, to let us know that we're moving in this horizontal direction, are the utricle. Okay, so these are the otolith organs. We're going to see they give us, all right, sensation on where we're moving in a vertical plane and a horizontal plane. Okay, the semicircular canals, they're going to give us rotational or dynamic movements. All right, so we'll look at these, but first we're going to look at the otolith organs, the utrica and saccula, that are giving us horizontal and vertical sensation. Right? So our cells of our vestibular apparatus, they are these hair cells. They are modified epithelial, and they have this stereocili along with this longer big kinocilium here. And you can see the kinocilium is tethered with these stereocili. These are the cells, and basically they're going to be moved depending on what the direction is taken. And basically similar in both organs, both the utrica, saccula, and semicircular canals. Okay, but we're going to look at how they differ in that aspect. They're housed a little bit differently. Their range, stereocili, are arranged in increasing height, and then the largest is that kinocilium. All right, and here is how they're activated. Remember, they are housed, there is endolymph. These sensory cells are housed in endolymph. And what is that high in? It's high in potassium. So if potassium channels open in these cells, then we're going to get potassium rushing in and depolarization. Okay. Movement is going to cause these cells to bend in a certain direction depending on which way the movement is taking place across them. What we are going to see is bending towards the kinocilium is going to open up potassium channels. These are going to be these mechanically gated channels. Remember, distortion causes, or stretch on them causes them to open. So mechanically gated potassium channels open when the bending of these cells heads towards the kinocilium. Right? So you can see at rest when the kinocilium is upright, no movement, we're just standing still. Okay, here is the firing action potential of those cells. Now when we bend towards the kinocilium and we open up those potassium channels, we can see those cells get depolarized and there is a greater amount of, or greater frequency of action potentials. And on the opposite, if I bend away from the kinocilium, what happens compared to at rest? It is much or less frequent, right? And so that's information the brain's getting. It's getting information from these, and what we're going to see is these are arranged in a different way. So one movement will cause some cells to go in this direction away from the kinocilium, and others will cause it to go towards this kinocilium. Okay, so bending towards kinocilium, depolarization. Bending away from kinocilium is hyperpolarization. All right, so let's look at the utrica and saccula. They are housed in these structures called the macula. You can see the macula, all right, here. This would make up the saccula because it's arranged in this direction, so it's going to give us movement in up-down fashion, where the utricle is set up in this direction, and it's going to give us movement in this direction. But both of these macula are housed in this otolithic membrane. Okay, this otolithic membrane, because it has otolith, it has these crystals, these heavier crystals, and this gel-like structure. You can see here are the hair cells, these purple things projecting out into this membrane here, and then that membrane has these heavy crystals on it, so it helps accentuate when the movement takes place. It's going to cause more bending because those heavy crystals are going to move in the direction of the movement. Okay. So here we go. Let's look at head movement in sideways direction. All right. And let's look at the otolithic membrane. We got the kinocilium. Okay. Here's my head. Here's the arrangement. And here's the arrangement of those cells. And we're going to see some of the cells in the otolithic membrane are arranged with the kinocilium on this end. Okay, they're not all arranged, the kinocilium on this end. There will be some arranged this way, so your brain's going to get multiple information. It's going to get some that are bending this way, but others that are bending in the opposite direction. The kinocilium is bending opposite, and so these will get hyperpolarized. So the brain can get multiple information. Hey, I'm getting hyperpolarizing here, polarization here, depolarization in these ones. Then it puts it together and gives you the integration of, hey, the movement must be in this direction. Okay, but let's look at this head movement. Remember, if the movement is towards the kinocilium, we're going to get depolarization, and that's going to cause increased firing of those cells. In this case, the kinociliums are lined up. The kinociliums are on this end. All right, so movement in this direction is going to cause depolarization. So bend my head, well, I'm bending left. That's going to cause movement in this direction. It's going to bend towards the kinocilium, and what do you see happening? An increase in firing. Whereas these kinociliums, when they're tilted in the right direction, all right, the movement is away from the kinocilium, all right, and remember here are those heavy crystals. It's going to bend in this direction, and we're going to get decreased or hyperpolarizing in the cells and decreased firing of the neurons, okay, or of the cells. Okay, so we have these arrangements. So we can get vertical, and we can get anterior. We can also get lateral. So we're getting just one plane of movement, though. Or backwards, boom, boom, boom. We're not getting a dynamic. We're going to see with the semicircular canals, we can get information on dynamic movement, where our body is when we're doing dynamic movements. Here's we're just going in one plane, lateral, vertical, a horizontal plane, either front, back, or right, left, up, down. All right, vertical direction, but in one plane of direction for these utrica and sacula. Okay. So for rotational, more dynamic movement, we have the semicircular canals. Movements that are rotational, where we're not just staying in one plane. The semicircular canal can give us information on which direction we are moving, which way the body is moving. Okay, so we have 3 of these canals, you see here, and they're lined up. One's lined up this way, one's lined up this way, and another's lined up this way. So they're at basically 90 degrees of each other. Where these are going to be housed are in these little swellings here, these ampulla. Okay, the crista ampullaris is where the sensory hair cells are going to lie. Again, it's going to be bending. This movement is going to cause the fluid to move. We're going to see we don't have the otolithic membrane, but we got these little cupullas and this indolent fluid, and the fluid movement is going to cause the bending of these cells. All right, so we got here, boom, 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 and instead of the otolithic membrane, we have this gelatinous membrane coating. It's like a little mushroom or a little cup in which the hair cells are housed in. Okay, and the utrica and sacula, we got the otolithic membrane, the otoliths. Here we got the cupula, and what's going to happen is movement, depending on which rotational movement, depending on which way we're moving, is going to cause the endolymph to bend the scapula. Okay. So let's take a look how this takes place. And let's say we are doing a pirouette, right? This is the one that is basically in this direction. Okay, it's rolling in this direction. And so when I go to move, that's going to move. My head is going to move when I do my pirouette. All right. The fluid is slow to move. So she's moving in this direction. You can see the fluid is slow to move, so the bending happens in the opposite direction because of the inertia of the fluid. When I start to move, my head's moving very quickly, but the fluid's not moving quickly, so it causes the bending of the cupula to be in the opposite direction of my movement when I start the movement. Okay? Then I stop, what happens? At a certain point, the fluid starts to catch up after the initial inertia, starts to catch up and starts moving around in the direction, in the direction that I'm moving. And when I go to stop, now the fluid is pushing the cupula in the opposite direction. So I'm getting movement of rotational acceleration. In lab, we would do this certain test where if you could get, what would happen if you got constant velocity? There would be no acceleration, and so there would be no inertia or fluid change. The fluid would be going in a constant motion, and we wouldn't have more bending. Okay, so we're getting acceleration. Other thing I want to point out here is we have these in 3 different planes. All right, let's look at, we got the superior. Right, here's the superior here. Okay, it's arranged in this direction, so the loop goes this way. So if I was to do a somersault, my head going over like this in this direction, that would move this fluid in here. That would get bending in this superior. Okay, the lateral. The lateral is for my pirouette here, spinning on my long axis, because it's arranged this way. It's arranged in this direction. And so when I go to spin, that's going to cause movement in mainly the lateral semicircular canal. And then finally, the posterior is set up basically this way. It's going like here from left to right in a loop. And so if I want to go do a cartwheel, that would cause movement mainly in the posterior semicircular canal. Okay? So the beginning of the movement, the fluid doesn't move here. My head's starting to move, but the fluid hasn't caught up yet, and it caused bending in the opposite direction of my movement. Right? Once I'm into the movement, I've accelerated, I'm into the movement, I'm continuing around, the fluid starts to move, and then when I stop, now my head is stopped, but the fluid is now moving, and we get bending in the opposite direction. Okay, so this is how we're getting our dynamic movement. So putting this all together to give us equilibrium, this information we're getting from the vestibular apparatus is just one component of the equation. Okay, the vestibular apparatus, that information is going to be sent to the cerebellum and then to brainstem, and the brainstem can then coordinate eye movements and control body movements depending on the information that it is getting. All right, but there are multiple components for this equilibrium. You got your eyes, information from your eyes, because when you're spinning, you're looking and you're looking at objects to see where you're at in the world. Also, you're getting information from cutaneous receptors. When I'm spinning, I can feel my pressure on my skin. changing. I can also feel the pressure or the stretch in my proprioceptors, in my joints and my tendons, or in my muscles. So all that information is being coordinated, sent to that portion of brainstem, and I can control the movements for eyes and my body parts so I don't lose my balance. Okay, so it's all coming together. Just let's rehash this before we leave. For equilibrium, here is those hair cells, right? They got that kinocilium. Remember the bending causes bend on the stereocili. We get the potassium channels opening. That causes potassium to rush in. Those are mechanically gated. So when the bending occurs, they're mechanically gated. So the bending causes the opening of the potassium channels. Potassium moves in, right? Bringing its positive charge. That causes our depolarization. And just like a neuron, when it gets depolarized, we get our voltage-gated calcium channels open, and boom, we get release of neurotransmitter to the sensory cell. That sensory cell can bring it to cerebellum, and there we get our information so we can get equilibrium and we can make our decisions on where we have to move via our body or control of our eyes. All right. The other information we're getting out of our inner ear, where our sensory cells are, we're going to get our auditory, our hearing. Okay, and that's housed in that snail-like structure, that cochlea. Where we're going to get the hearing, where we're going to get the sensation. We're going to get basically what's causing what's our stimulus is sound waves. Okay. The sound waves are going to cause fluid movement. We translate it into fluid movement in the inner ear, and then we'll get the bending. Again, we're going to have stereocili, just like we did with our vestibular apparatus. We're not going to have the kinocilium when we get in the cochlea, but we still have those hair cells that the sound waves are ultimately going to be translated into bending these hair cells, these stereocili. Okay, our sound waves that move through the air. Two main components of it is we have the amplitude of a sound wave. Okay, that'll give us what we call intensity, how loud it is, right? Depending on how high the wave is. The other characteristic of the sound wave is we have frequency. We got our wavelength. We have a smaller wavelength, say like this, right? From apex to apex. All right, this red would be a higher frequency than the blue. All right, and we're going to see our frequency can give us our pitch. High. All right, so our frequency is going to give us pitch. Amplitude is going to give us intensity or decibels, how loud it is. Okay, those are the two components, but we're going to see how. First, we got to translate the sound waves into fluid movement into the inner ear to get the bending. The fluid movement is going to cause the bending of the stereocili. So our little anatomy lesson, because remember we got sound waves. They're in the outside environment, in the air out here. We're going to translate those into movement into fluid. So we have three regions, our little anatomy lesson of the ear for the day. We got the outer ear, middle ear, and inner ear, where we have the cochlea. But this middle ear and the outer ear are going to help us translate those sound waves into fluid movement in this cochlea. All right, let's look first. Sound waves are coming in here. All right, we have the auricle, we got the ear. the outside ear that you know of. External acoustic meatus. So the auricle is going to funnel the sounds into the external acoustic meatus. Your ear canal, right? And it's going to funnel the sounds onto the tympanic membrane, or your eardrum, commonly known. Okay? And that's going to cause the sound waves hitting the tympanic membrane is going to cause vibrations on the tympanic membrane. Tympanic membrane is basically the border between the outer and the middle ear. Okay. So here is that tympanic membrane. We're getting vibrated, and you can see there are those three bones. Again, our little anatomy. We got the malleus, the incus, and the stapes. Malleus is looks like that little alien baby that's crawling on the tympanic membrane here. Right? It's connected to the incus, and then the stapes. Incus is connected to the stapes. The stapes is connected here to the oval window. That is what's going to lead in to the inner ear. We're going to have perilymph and endolymph, where we're going to get movement into that fluid, and we're going to get fluid movement via the actions taken here. So when this vibrates the tympanic membrane, it's going to cause movement of this, of the malleus, and the malleus connected to here, and basically the vibrations that happen here are going to occur over here, but the key is through this process here, we're actually amplifying the sound waves. Okay. If anybody's ever had a inner ear infection or middle ear infection, you get fluid filled in here. This is air space, and so this is allowed to move freely. You get fluid filled in here with an infection, and this isn't allowed to move as freely. That's going to slow down the vibration, and so what happens many times when you have middle ear infection? You're like, I had trouble hearing, because you're affecting the movement of these middle ear, the isosceles, okay, or sorry, ossicles. Right, so we're going from large to small when we go through the middle ear, and so we're getting amplification of the vibrations here at what we call the oval window. The stapes is connected to this oval window, right, and that's going to lead us into the cochlea, lead us into the inner ear. All right, so the inner ear, the cochlea, the snail-shaped structure we're seeing here. If we were to slice it down, you can see it is this continuous. So this would roll around here and up here and roll around into three turns. And in these turns, to these canals, there are three basically canals in here. There's the scala vestibula, right, looking here, and the scala tympani, the below structure here, right, and housed in between them is the cochlear duct. This is where we're going to find the organs for hearing, the hair cells, the sensory cells, right? The scala vestibuli and the scala tympani, the bigger purple structures, they have perilymph, which is similar to the interstitial fluid. Where in the cochlear duct, where the hair cells are going to be housed, we have that endolymph. Again, that endolymph is high in potassium. All right, so the sound waves come in, and you're going to see the movement here is going to cause vibrations. It's going to move the sound waves into the fluid here in the scala vestibuli. So here is a more blow up of the organa corti, of the spiral organ. Okay, that is here. This is where that hair cells are going to be housed. There's that scala vestibuli, scala tympani, and the cochlear duct is here. Okay, we got our endolymph, our high in potassium, housed in this cochlear duct. Okay. The hair cells, they are attached to the basal side here. Okay. There's a basilar membrane, makes up the bottom of portion, the duct here. Stereocili, then project up. Right? Again, there's no kinocilia. And the hair cells are embedded in this upper tectoral membrane. Okay, kind of like what was in our macula and scapula. They're in the otolymphic membrane. The hair cells aren't just floating around, they're in that otolymphic membrane. And the semicircular canals are in that cupula. And here, these hair cells are embedded in this tectoral membrane. All right. So let's see how this translates to movement in our cochlea. First, here's a little bit better blow up of our organa corti, where we have our hair cells. These are our hair cells. There are outer hair cells and the inner hair cell here. The inner hair cell is the actual sensory organ for hearing. The outer hair cells are actually going to be there to help amplify the signal and movement to cause more bending in the actual inner hair cells. Okay, but this row of inner hair cells is what's picking up the sound waves to give us the auditory sensation. Okay, and remember they are housed in this tectoral membrane, and they have the basilar membrane in which they are embedded on. Okay, but here is that cochlea, and you can see in this scala vestibuli, that's this channel here, is continuous with the scala tympani. So the sound waves come in through here, come in through the oval window, and then move out through the round window. Okay. There is the stapes. Remember, the stapes is pushing in and making those vibrations to cause the waves to move into the fluid of the cochlea. Okay. You can think this is simplified because this actually would go in 3 turns. Okay, this whole structure is basically, if we took the snail and unraveled it, this is how it would look, but this is actually going into those 3 turns. Okay. One thing I want to notice is, if you remember our sound waves, we had 2 components or characteristics. We had the amplitude, okay, the height of the wave, that's going to give us how loud, but we also had the wavelength, known as frequency. Shorter wavelength, higher frequency, longer wavelength, lower frequency. Okay, that's going to transpire to our pitch, high versus high. All right, and you can see we have a higher frequency. This red wave is higher frequency. Okay, say 20,000 hertz, where this green is a medium frequency, 200, and then our low frequency is our blue wave. And what's going to differ is, where are these going to cause the bending? So this is how the brain's going to be able to interpret the pitch of the sound, in that these red waves, where does it cause the torquing of the membrane? You can see the red is causing it here, the green is causing here, and the blue is causing it further down. So depending on where in the cochlea, where in the ducts are we getting bending, the higher frequency is going to cause bending of the cells earlier on in the duct, where the medium will be a little bit later, and then the low frequency will bend the cells later in the duct. Right? And the brain's interpreting, hey, I'm getting information from these earlier stretch here. I'm going to interpret it as high frequency. getting signaling, higher signaling from the cells here, I'm going to get medium frequency, and I'm getting bending of the hair cells, signaling from the cells later on. I'm going to interpret it as lower frequency. Okay, so that's how the brain is going to be interpreting frequency depending on what cells are getting stimulated, where they are in the cochlea. Okay, where they are in those turns. Okay, earlier turns, higher frequencies, medium frequency, and then later turn going to be the lower frequency. This side shows us how we're going to get activation of the hair cells. So the movement, those waves coming in are causing movements in the basilar membrane. Okay, they're coming down and up, and when the basilar membrane gets moved, okay, we're housing those tectorial membrane, and it's going to cause a shearing force on the tectorial membrane to cause those hair cells to move, depending on which way the wave's moving in, coming up, going down. Sorry, going down or coming up. It's going to depend on the shearing. All right. And we're going to get bending of the hair cell. Remember, there's no kinocilium here, but the bending is going to do basically the same thing in that the bending, there are mechanically gated potassium channels. Potassium channels open when there's bending, and we're going to get movement of potassium into the cells. All right. So activation, bending of the hair cells towards the high end. That's going to open up my potassium channels. Right. I'm high in potassium. That'll cause potassium to move in, cause depolarization, and movement of calcium in to release the neurotransmitter from the inner hair cells, and that will activate the sensory neurons that will bring the information, start the pathway back to the brain to get integration. All right. So strength of stimulus. Okay, here are our potassium channels. You can imagine the bigger amplitude, okay, say this compared to this, which is going to cause more bending of these hair cells. Yeah, the higher amplitude. So higher amplitude is going to bend it more. I'm going to have more channels opening. I'm going to have more influx of potassium. I'm going to have a stronger signal, and I'm going to have more neurotransmitter release and more frequency of signal. Okay, so this is going to cause higher frequency of signaling compared to, say, the lower amplitude. Okay, higher frequency, not frequency of the sound wave, but higher frequency of stimulation of these cells, causing more neurotransmitter to release. Okay, higher frequency of action potentials, brain can perceive it as louder noise. Problem with very loud noise, though, is what's going to happen. You can see Mom was kind of correct when you went to the concerts. Hey, you're going to damage your ears. You're going to lose some hearing because these are nice little sensitive cells. You don't want to torque them and get them. Okay, you can damage them. Okay, by too much torquing. Loud sound's going to cause lots of bending on these hair cells. You get enough, you can have damage to them. All right, but again, higher amplitude, more bending, more signaling, higher frequency of action potentials set. Okay, lower amplitude, lower bending, lower frequency of action potentials. Okay, and so that's how your brain, how frequent the signal is going to give you how loud and where in the cochlea, which of these cells, where in the cochlea are these cells being stimulated. Earlier on, higher frequency, higher pitch. Later on, medium frequency, medium pitch, and further down the line, lower frequency, lower pitch. So we're getting both pitch and loudness. Okay, so high pitch earlier on in the cochlea and higher amplitude, so higher frequency of action potentials caused by it. High. Low pitch, lower on in the cochlea, further down, and it was softer, so it would be lower amplitude and would cause less bending and less frequency of action potential. All right, so that is the information the brain's getting, making interpretation of both pitch and loudness to it. So neural pathways, once we get the stimulation of those hair cells, we run through the vestibular cochlear nerve, through the brainstem to the inferior cochlea, but then where do all good sensory cells go? Thalamus, and then the thalamus can spice it out to the auditory cortex, which we see over here. Okay, you see the auditory cortex. Remember, the somatosensory cortex was somatotopic. Certain regions of the body go to certain regions of the somatosensory cortex. Well, tonotopic means certain regions of the cochlea go to certain parts of the auditory cortex. And this is where we get kind of a mapping for pitch, for the different tones. You can see those earlier on, right, would be the higher frequency. So higher frequency would go to this portion of the auditory cortex. Remember, higher frequency waves are going to stimulate the cochlear, the hairs in the cochlear duct sooner on than the lower frequencies. Okay, whereas we go further along, right, the medium tones or medium frequency would stimulate the cells through here, and they would go through this region of the auditory cortex, and those with the low tone are going to stimulate the cells later on in the cochlear duct, and those will go to this region of the auditory cortex. Right, so where the brain's getting information, if it's getting stimulated here, there, or there, it can determine and give you a perception of pitch. All right. So how can hearing be affected? What locations can we get hearing damage to, or damage so we get loss of hearing? We got 3 categories to deafness. We can have dysfunctions in conducting the sound waves, sensory neural being the sensation, mainly that's going to be through those hair cells being damaged, or we can have central deafness. Okay, part of the central nervous system is being damaged to get the loss of the hearing. So conduction deafness is where you see your classical hearing aids, right? They're blocking in the external meatus. You got a bunch of wax buildup. That's going to affect, we talked earlier, the middle ear infections. You get that, those ossicles get fluid around them. It's going to prevent them from moving. All right, how can we fix this? Well, one's getting rid of the middle ear infection, but we can also amplify the sounds using those classical hearing aids. Okay, the other second, sensory neural. This is where damage comes to those hair cells. You've gone to rock concerts all through your youth, and you've damaged your sensory cells. Right, you're not going to be able to sense those sound waves coming in. Right, or if there's just genetic damage to those hair cells, but the actual hearing works fine. The pathways to the brain work fine. You can actually stimulate those by using those cochlear implants. Okay, stimulate the dendrites as long as they are still viable, as long as the pathway back to the brain is fine. You can still use the cochlear implants to help improve the hearing. Now, central deafness, you get damage to the cortex, you get damage to that auditory cortex, or the pathway, the neural connections to the cortex from the ear. Then there's not a whole lot you can do because you're not going to be able to perceive the sounds. All that other stuff, the hair cells could be working fine. You can conduct them in. The hair cells get bent. But if you have damage to the pathway going back to the brain or the actual part of the cortex that puts perception, you're going to have loss of sounds because you're not going to be able to perceive it. Not a lot you can do in that case. But for the sensory neural, we can get the cochlear implants, and for the conduction, you can use those traditional hearing aids where we're just amplifying the sounds. So that's it for hearing and what's taking place in the ear with our equilibrium and hearing. Now we're going to move into the most complex of the sensory or the special senses organs. And we got the eye. We have a lot that has to take place for us to get our vision, to get our visual sensation. So now we're going to get into, we're in the sensory physiology with the most complex of the special senses in vision. Here we're looking at light waves. We're looking at light waves of different wavelengths from the electromagnetic spectrum. And those light waves are going to activate our photoreceptors. We have photoreceptors that are stimulated by this light wave. Okay, there are many different light waves. We got things we've probably heard of: X-rays, ultraviolet from the sun, infrared, like our remote controls, microwaves, and even radio waves. Okay? But why don't we see these waves? Why don't we see the radio waves coming into our car, to our radios? Why don't we see microwaves when we turn it on, or see X-rays when we're getting X-rays taken? It's because our photoreceptors are not stimulated by those wavelengths. They are only stimulated by the wavelengths in this visible light range. Okay, this 400 nanometer to this 700 nanometer, this blue to red visible light range. Okay, those are what are going to stimulate our photoreceptors, and therefore that is why we can see these lights. Okay, the visible light. Okay, this is most complex in that for our vision, we have to have a lot to go right. We have to have our eyes focused in the right location. We got to be able to bend, whoops, where are we at here? Bend the light waves properly so they land on the right part of the retina. And then we got to be able to properly carry that information back via our nervous system. Okay, so a lot has got to go correctly so we can get proper vision. Right, let's get into the anatomy, our anatomy lesson. What we're going to be using the most, we're not going to be worried so much of the sclera, choroid. Where we're going to find our receptors are, you remember, the retina. Okay, we got the retina here, and this is where our photoreceptors are going to lie. This is where we need to refract the light, where the most high density of photoreceptors, of cones, we'll talk about cones and rods, but our ones for high visual acuity are cones. They lie in this fovea centralis. Right, so this is where we're going to want to focus the light that we need. Right, the axons from our neurons, from our neural layer of our, run through here into our optic nerve. Okay, give us the optic disc that's back here. All right, and the front of the eye. Front of the eye is going to house a lot of the structures that maintain how we refract the light and how much light is actually allowed into the eye. So if you remember, we have the outer clear portion, like an extension of the sclera, the white, that is the cornea. Okay, this is where the light's going to come in. All right, we got the iris, that's the colored portion of the eye, and it is actually a muscle that controls the hole there, that pupil, and that's going to help control how much light is coming in to the eye. All right, we have a ciliary body here. All right, with the zonular fibers and suspensory ligaments that are connected to this big structure here, which is the lens. Right, this is going to be another structure important in controlling the refraction of light. And the light is, or the eyeball is not air filled. Right, we have in the frontal chamber here, we have the aqueous humor, and then in the back we have that kind of gelatin. If you guys had anatomy class, you might have dissected a cow eye. If you had anatomy several years back, since most classes haven't been in with COVID, there's this gelatin structure that fills this eye and actually holds this retina into place. All right, and that gelatin is that vitreous humor. All right, so we're going to need this stuff up here properly working so we can get the proper refraction of light, control the amount of light, and ultimately we want to get the light that's coming into the eye focused back near this fovea centralis, this region of the retina that has our lots and lots of receptors. If we have lots of receptors, we can get much better acuity, much sharper vision. Okay, so we're going to go through and how all this works and how we get this focused and how we get the process back to the central nervous system to get our perception of vision. All right, just a closer look, looking at that ciliary body. Okay, that's a ciliary body with this little suspensory ligaments. And what we're going to see is it's attached to this lens, and that's going to allow us the lens to be pulled on and stretched, or to be relaxed, and the lens can get more bubblegum-like, more round. Okay, we got the iris up here. It's going to control this pupil here, the size that's allowing the light to go through into the retina. All right, let's first control the amount of light that's getting allowed to go through the eyeball back to the retina, right? Because we don't want our pupil, that's the opening between, right? That's controlled by these muscles of the iris. You don't want it too open, letting too much light in if it's bright light, because it's going to bleach out the receptors. That's where you get blinded by the light. Too much light's getting in, you can't see. But on dim light, you want to have the pupil large because you want to have more light in so you can get enough light in there so you can get discrimination between the reflected lights, right? So you can get some contrast. So in dim light, we want it big, and in bright light, we want the pupil small. All right, and how do we do that? There are two muscles that make up the iris. There are these radial arranged smooth muscles, these here, that would pull pupil in this direction and open it up. So the radial muscle, when relaxed, when contracted, it pulls the pupil open. When it's relaxed, it's allowed to allow the pupil to contract and get smaller. Right? The circular muscle, on the other hand, that's the inner circular muscle here. When it contracts, right? When it contracts like this, and will make the pupil smaller. So you can see these muscles are working almost antagonistically. All right. So when one is contracting, the other has to be relaxing. And so we have two systems. Our autonomic nervous system, because it is smooth muscle, our autonomic controls things we don't have to think about, the involuntary stuff, which smooth muscle is one of them. We have the sympathetic and parasympathetic controlling these muscles. In dim light, the sympathetic nervous system, okay, is going to contract the radial arranged muscles. Okay, so this contracts usually when the sympathetic is activated. The parasympathetic, which controls the circular muscle, is dampened down, right? And so we can get the pupil opened up. Remember, the sympathetic is your fight or flight, so think the radial. What happens when you get scared? You got those big old pupils looking out for danger taking place. All right, now in bright light, we want our pupils contract. We want them narrower. We want a small pupil. We won't want too much light in. And the parasympathetic is going to be activated, and that contracts the circular muscle here. When the parasympathetic is activated, usually the sympathetic is dampened down, so these muscles will relax while the parasympathetic is contracting the circular muscle to control the light. All right, so that's one of the first controls. We've got to control the light so we can make sure there's not too much and not too little, depending on this is what's taking place, whether it's dim or bright light out there. All right, so refraction. Right, we're seeing light being reflected off of things, and the light is coming in the eye. And depending on where the light is coming from, whether it's coming from far or coming from very close up, depends on how we need to bend the light, because we have to get the light bended into that right location, into that fovea centralis is where we want to get the light sent to. And so we have to get it focused properly, and so we have to properly do what we call refract the light that's coming in. Okay, you can see here, it's coming in this direction. We got to refract it, and you'll see when it gets refracted, the image is going to be inverted. Okay, but what structures of the eye cause refraction? Right, the majority of the bending, or that outer clear portion of your eyeball, the cornea, can control the majority of the bending. Okay, it's going to bend. The key with the cornea, though, is we can't change its shape. Okay, unless you get laser surgery, and that's what you're doing with laser surgery, is you're changing the structure of the bending of the cornea, right? But we can't change it. We don't have a physical means. The lens is where we can adjust the bending. Okay, this is the most bending. It's number one. Number two is the lens. Cornea, we cannot adjust, but the lens we can adjust, and we can change by depending on where the light waves. Because when we're coming from afar, the light waves are coming in here. We don't have to bend as quickly. Whereas when the light's coming in, okay, or sorry, from close up, the light's coming in farther out, we have to bend it much quicker. And to be able to make those adjustments, to be able to refract differently, we can change the shape of the lens to change the amount of refraction. Again, it's the autonomic nervous system. We don't have to think, hey, I got to bend the light more. The autonomic nervous system is controlling the shape of the lens to allow us to properly refract the light. Okay, another can't control, but the humors, those fluid fields, kind of like in a water, right? If you're underneath in a pool and looking up, what's going to happen? The water's going to bend the light. Okay, so the humors add some bending, but they would be the least amount. So top bending, most bending cornea, lens is second, humors third. Cornea we cannot change. Well, cornea and humors, we can't adjust the refraction taking place, but with the lens we can adjust the refraction. Okay, let's see how this operates. Get in. okay, for the lens. We can adjust it. We have these ciliary muscles that are here. When the ciliary muscles, so the ciliary muscles, round in shape, when they relax, okay, and they are connected to these suspensory ligaments that are connected to the lens. Okay, the lens is elastic in which it can be pulled, brought back. All right, we can pull on it, stretch it out, and when we decrease the pulling, it will bounce back. It has this elasticity to it. It is flexible. Okay, when the ciliary muscles are relaxed, all right, the lens gets more stretched. All right, and when it gets more stretched, we're not refracting as quickly. All right, so for coming in at a closer distance, when we're seeing far, you can see the light is coming in this distance apart. We don't have to refract as quickly when we're looking at a distance, right? And therefore the lens can be in a more stretched shape. Okay. So here's that taking place. Light rays distance are more parallel, so don't need as much refraction when we're looking at things at a distance. Okay. When we're looking at stuff closer up, they're more divergent. Okay, you see this is much further apart than, say, here. Now when we're looking at things closely, we got to get the refraction going much quicker. We got to bend it very quickly. All right, and to do that, we want the lens in a more round shape. And so when the ciliary muscles contract, that takes off a lot of the stretch, the pull by the suspensory ligament. We contract the ciliary muscles, and now it can bounce back. It's elastic. All right, the lens bounces back, and it is in this more circular structure. Okay, and that is going to allow us to bend the light rays much quicker. Allows us to refract them much quicker. Okay, so you can see we have to adjust the light coming in, and we also have to adjust the refraction properly to make sure we get the light reflected in to the right direction, or the right refraction, so we can get it to where it needs to go. Okay, this is called accommodation. This bending of the lens that allows us to help focus the light properly, to get it bent properly so we can get it focused into the right area of the eye. Okay. Problem is when you get old like your professor, what happens? All right, do you think this lens is as elastic? No, things get old, they get old and crusty, as my kids like to call me now, old and crusty, but the lens isn't as elastic, so it won't bounce back into as round a shape, and I can't bend. If it's not as round, I can't bend it as much, and what happens? All right, I lose my close-up vision. So that's what's happening with presbyopia. It's when you need, now I need reading glasses when I'm looking at my phone or so forth. Okay, I get presbyopia. This doesn't get as round as it once did in my youth, and I can't bend the light rays as quickly, and therefore I can't get it focused where it needs to, and therefore I need the glasses to help me bend this, start bending the light rays even before it hits my lens. All right, so I can get the proper refraction back to the retina. All right, disorders in focusing. All right, those that are nearsighted, can't see far, they would be called have a myopic eye. Normally it is a long eyeball. the eyeball is too long, and you can see where the focal point happens in somebody with myopia. The focal point ends up short. We want this to be back here. We want it to be focused. So the lens can only accommodate so much. If the eyeball is too long and we get this focusing, the lens, even though it's getting stretched, okay, or getting it to accommodate, I only can stretch it so far, so I can't get it focused in the right location, right? The lens can only do so much, right? If my eyeball is too long, it can't compensate, it can't stretch out even more to allow this to take place, and so therefore I need, what do I need? I need a concave lens. The concave lens is actually going to divert the lights out, right? And so when it the concave lens allows bending to be further back, and now I can get the focal point where it needs to be. Somebody with a hyperopic eye, they are farsighted. Okay, the eyeball in this case be too short, and the focal point's behind the retina, not in front like here, but behind the retina. Right? How do I get this? I got to bend it even quicker. Remember, the lens can only compensate for so much. Now the eyeball's too short. I can't get the lens even rounder to bend it even quicker. So what do I need? I need a convex lens that starts the bending already of the light before it even hits the lens, and now I can move that focal point up to where it needs to be. Right? So that is what you're doing with your glasses. Most of us have bad vision. Usually it's nearsighted. Okay, but what happens when you get older? Right? It's not so much the eyeball, it's the lens with the presbyopia can't get as round, and now I need bifocals to help me do this end, but I still have a myopic eye that when I need to see far, I need the lens to do that. All right, so those are disorders in focusing. All right, so now we've controlled the light, we've controlled the refraction. And where are we sending this to? We're sending it to the retina. Okay, so 2 layers. We got the pigmented layer, right? That is this layer down here. Right, and so where we're looking at here to keep you in is the eyeball, or the cornea is out over here. So light is coming in this direction. Right, so these layers here are closer to the light source coming in from the eye, and this is further back. This is in the back of the eye. Right, so the pigmented layer is down in here. What's it going to do? It helps absorb light energy. Pigments are absorbing light. We're going to see our photoreceptors have pigments in them that help absorb the light, and that's what's going to cause the stimulation of our photoreceptors. Okay, our pigmented layer is associated with the photoreceptors. We're going to see they're almost like astrocytes, and they are support cells for these photoreceptors. The ones that are going to respond are going to transduce the light energy. Okay, the neural layer is here. We got the photoreceptors, the bipolar cells, and the ganglion cells make up the neural layer. Okay, what's going to happen is light's coming in. You can see light actually has to run through this neural layer this is where the light energy is going to be picked up by the photoreceptors. Okay, so we actually have to run through the neural layer, all the way through the neural layer to the back portion of the neural layer to get activation of the photoreceptors. Okay, once they get activated, we're going to transmit to the bipolar cells, then to the ganglion cells, and then the ganglion cells' axons are what become the beginning of the optic nerve. Okay, so light comes in this direction, the signaling pathway goes out in the other direction. All right, so let's look at the layers a little bit closer. All right, 3 distinct layers to the neural layers. We already said they're photoreceptors, and they're going to come. Our photoreceptors are going to come in 2 varieties. They're going to come in rods and cones. All right, rods are very sensitive to light, so they can be activated in low light. When we're at dusk and there's very dim light, we're activating more of our rods because they are sensitive to that low amount of light. Okay, they are not sensitive to color. Right? Do we see bright colors when it's dusk out? Do we see all like these bright colors? No, it's more like gray tones when it's dusk out because we are utilizing the rods. Our cones are less sensitive to light. They need bright light to be activated. Okay, but we can get color. Red, blue, and green are going to be our colors, and any combination of those are what's going to give us our color. When do we see nice and visual bright colors? Is when it's very bright out, because the bright light allows us to use our cones and gives us the high acuity, very sharp vision. Our vision is very sharp when we're using cones. When we're using rods, it's more blurry and kind of fuzzy. So the flow, though, we get these activated. The photoreceptors then talk to the bipolar cells, and then the bipolar cells talk to the ganglions, and then the ganglion axons actually become the optic nerve. That's what's going to send it back to the central nervous system. Okay, making up that cranial nerve too, right? So let's look at our rods and cones. All right, there are basically 2 segments to these rods and cones. There's the inner segment. All right, inner segment is this portion here, axonic end with the nucleus. Okay, it's got their nucleus. It's got your organelles. This has got all those features of your normal cell. These outer segments are what contain the region for reception, where we're going to pick up the light energy. All right, you can see our cones much smaller, outer segments, but you can see they have these discs in here. And these discs help to absorb the light. Okay, they contain the photopigments. Your pigments absorb light, and it's going to be the absorption of light in these pigments that are actually going to ultimately result in the signaling of these photoreceptors. Okay, again, these discs help in trapping the light, and they'll be continuously added and removed by those helper cells of the pigmented layer. Okay. So the pigmented layer, before we get into the actual signaling taking place in here, you can see they have many functions that help operate these photoreceptors. Okay, you can see supply nutrients to photoreceptors. They're going to help absorb scattered light. So there's light coming in, some of it is scattered. There is melanin in there, so it helps absorb the extraneous light so we can get the light we need focused on there. They help phagocytose the discs and the receptors. They also recycle the pigments Okay, we're going to talk about the pigments in a bit because those are going to be what's signaling or what's going to cause the signaling in the photoreceptors. Okay, kind of like astrocytes stabilize ion composition so these cells can respond properly. So they're almost like the astrocytes, but astrocytes of the photoreceptors. Okay, they're kind of the gophers to help make sure these photoreceptors can operate properly. All right, so let's get into our signaling. All right, our rods. We'll look at how the rods work, but basically the signaling in the cones is very similar, just going to be different wavelengths of light that's operating. All right, so we're looking specifically at the rods. You can see the absorbance in the rods. There is the pigment, and this is what difference between, say, the rod and the different cones are the different pigments that are used for absorbing the different wavelengths of light. In the rods, we have rhodopsin. Okay, you can see where they absorb. Their highest absorption is around 498, almost like a purple or a light green. Okay, absorbed in the green, blue-green region, purple region. You'll see at night when we're using our rods, one of the colors we can see best is green because that is where color lies. Okay, green light lies right around that area, so we can see it more easily at night. Okay, but again, this is what the different pigments in the different cones and different in the rods, you can see they absorb, the pigments absorb different wavelengths of light. Okay, we're focused strictly on here right now, but we'll relate this. The signaling is going to be similar between the rods and the cones. It's just that they're going to be stimulated by different wavelengths of light. Okay, so in our rods we have our rhodopsin. Oh yeah, here's where it gets kind of tricky. Okay, the rods, the photoreceptors work a little differently. Most of our sensory cells, what has happened? They are at resting, but then when they get stimulated, what happens? They get depolarized and they start sending a signal and releasing neurotransmitter. In our photoreceptors, the neurotransmitter is actually being released when they're not stimulated, when they're in the dark. Neurotransmitters being released by the photoreceptors. So that's the case. What do you think the neurotransmitter is? Do you think it's an excitatory or an inhibitory neurotransmitter in the dark? Since they're activated in the dark, they're actually inhibiting the pathway, so it's an inhibitory neurotransmitter. So what we're going to see is to get stimulated, we're actually going to hyperpolarize the cell and stop releasing that inhibitory neurotransmitter, and now we can get signaling. Okay, but let's see how we get from a depolarized cell in the dark to hyperpolarized in the light. Okay, and this has to do with, here are those discs, right? We have those discs in the photoreceptors. Here we're looking at rods, so we're talking about rhodopsin. Okay, rhodopsin is here, is going to absorb the light. In this rhodopsin, there is this retinal molecule. In the dark, the retinal molecule is in what we call 11-cis transformation. Okay, look at it. Difference here is it's an all trans retinol, you can see there is just a small change in the structure down here. Okay, but in dark, the retinal is in 11 cis. When it is here, you can see there are G proteins coupled here. They are inactive in the dark. Okay, there is this enzyme, phosphodiesterase, is also inactive in the dark. Okay, what does phosphodiesterase do? It actually converts cyclic GMP to GMP. But since the phosphodiesterase is inactive in the dark, the phosphodiesterase is not converting cyclic GMP to GMP, and so cyclic GMP levels are high in the dark. And what function does this cyclic GMP serve? The cyclic GMP binds and keeps these sodium channels open. So in the dark, we have lots of sodium coming in. And what does sodium do to our cells? We're back to regular interstitial fluid. We're not in endolymph and high potassium. There is high sodium outside and low sodium inside now. All right, so sodium rushes in, depolarizes the cell, and that causes the cell to be activated and release its neurotransmitter. Okay. Again, it is an inhibitory neurotransmitter. So in dark, we are inhibiting the pathway. What happens when light comes in? The right light energy. Since we're talking rhodopsin, we're saying around 500 nanometers for wavelength. So that 500 nanometer light is coming in, all right, because this rhodopsin will not respond to those light waves further off from 500, all right? It only responds to those that are not exactly at 5, but we have a little range. It's going to be most active at 5, but going down to say 400 to say 600, it's going to activate a little bit, but it's mostly activated at this range. And by activated, I mean at 500 nanometers, that 11 cis retinol is going to be converted to all trans retinol. And when that takes place, that activates that G protein. All right, now the G protein is activated, and the G protein goes, or cascades, goes and activates that phosphodiesterase enzyme. Right? What did phosphodiesterase do? It converted cyclic GMP to GMP. Now cyclic GMP levels are low. Cyclic GMP was holding my sodium channel open to allow sodium in. Now with sodium not being allowed in, my cell can hyperpolarize, and now I'm not sending neurotransmitter. It cuts off by hyperpolarizing. Now I'm not serving neurotransmitter. Now I'm removing the inhibition on the pathway, and now the pathway, the neural pathway, can signal. Okay, so it's a little reverse. In dark, photoreceptors are releasing neurotransmitter. In light, they stop releasing the neurotransmitter, and then the rest of the neural pathway can signal. Okay, let's see. All right, so let's see how this works. We'll put the photoreceptors in the process with those neural pathways, right? In dark, right? That cyclic GMP is high, the sodium channels are open, and my cell's being depolarized. Okay, I'm releasing neurotransmitter, but it's inhibitory. So these cells signaling in these cells are put off because I'm inhibiting the pathway. In light, we go from 11-cis to all trans. That activates my phosphodiesterase, my G protein, and then my phosphodiesterase. Now cyclic GMP levels are going low. That shuts down my sodium channel, and now my cell is hyperpolarized. It's inhibited. But when inhibited, it will not be sending there's no inhibitory neurotransmitter being sent to the bipolar cell, and now these cells are allowed to fire. The bipolar to the ganglion, now we get our signaling. Okay, so that is in dark. Our pathway is inhibited by this inhibitory neurotransmitter being sent out. In light, the inhibitory neurotransmitter is not being sent, and now I can signal through my pathway. All right, so what we looked at was what's signaling through the rods. The signaling is very similar in the cones. The difference is the wavelength in which the cones are stimulated by. Instead of rhodopsin, the different cones, depending on which cone it is, has a different photoopsin. Okay, there is a blue cone, or what's also called the S cone for the short wavelength, S short. Okay, and you can see the wavelengths in which the blue cones are stimulated by. Those pigments are going to pick up that color. It's going to change the cis to trans when light of this color is coming into the blue cone. All right, there's M cone, and the photoopsin for M cone will pick up lights in this wavelengths. Right, M for medium wavelength, and then the red cone has a photoopsin that is going to pick up light in these wavelengths for long, L for long wavelength. Okay, but the signaling is the same. That light's coming in, it's turning that cis to trans retinal, all trans retinal, and now we get the activation, and this inhibitory neurotransmitter will stop being sent, and now those neural pathways can be sent. Again, these are cones, though. They're working in daylight. They're working in very high light environment, very bright light environments. Okay, because they are not as sensitive as the rods to light. There has to be more light to activate these cones than there does for rods. Rods can work in low light situations. Okay. So how do we put this together? This is what we call trichromatic, 3-color vision. All right, by what cones are being stimulated and by how much we can merge those signals together to give us the various colors that we see. So how does this all operate? It all operates on the brain can get different signals depending on where the wavelength is. You can see it's going to get multiple signaling, but you can see certain cones are firing more frequently or higher frequency at certain wavelengths than the other cones are. Depending on the amount of signal the brain is getting, it can put a perception on what color is. Depending on what the signaling is between all the cones, I can get higher. So in blue light being reflected, mainly this is going to be activated. Maybe we're down here and a little bit of this, okay, but very little red here. This would put us kind of in the cyan range. We're going to get blue and green together. So depending on the firing it's getting from these various cones gives us what our color is going to be. Your brain can put perceptions to the different firing rates and give a perception of what color it is by using these 3 different colors. So let's look at where these are located. Looking at our macula lutea, that is this bigger circle here. Okay, this here. And in the middle of the macula is this little pinpoint. Okay, the fovea centralis. And what do we see in this macula lutea, and specifically at this fovea centralis, is there's highly pigmented area. We're going to have lots of cones there. Okay, highest density of cones. Okay, we're going to be able, because cones are going to give us very high acuity. We're going to be able to make very fine distinctions in vision with activating this areas with high density of cones. Okay, almost no rods there. The rods are more out in this region, the outer peripheries. And so what are we going to get at this fovea centralis specifically is the sharpest area of vision. So where do you think you want to focus your light? Yeah, especially in bright light, you want to focus out here at the fovea centralis because you're going to get nice, highly acute vision. So, since that is your brightest region, your eye is trained. Your eye muscles are trained and your refraction is trained to focus that light on the macula and specifically the fovea centralis, right? Your eye muscles, your brain doesn't have your muscles focus, so the focal point is far away from that fovea centralis. You're focusing that light there. So in macular degeneration, where there's degeneration, those cones have been damaged. All right, you're in a normal person vision, right? Your eye muscles and your lens are all going to focus that energy at that fovea centralis. So you see that dot, right? Again, in macular degeneration, there's been damage to those cones in the macula around that fovea centralis. Your eye is still going to have the light focused onto wherever you're looking at, onto the fovea centralis, and what's damaged areas in the macula around the fovea. So therefore you don't see any vision there because this is where that damage has taken place to the macula, and you don't have cones there. If you don't have cones, you're not receiving the visual input. So you see that as a grid there. Okay. So why are we getting such visual acuity at the fovea centralis? Right, we have a high density of cones there. All right, so we can pick up the receptive fields for each of those cones are going to be smaller. And what else? Usually the light has to run through the neural layer to hit the photoreceptors at the back there. In the fovea, you can see this little indentation. All right, what's happening is that neural layer is almost parted. So that light is going almost directly onto those cones that lie in the fovea. We get that bright light, we're going to be able to get brighter acuity. Okay, besides the cones being high density, what else is happening with our cones is you can see there are 120 million rods and 6 million cones. There are only 1.2 million axons that are coming out to form this optic nerve. So what's that mean? That means that these rods and cones, there has to be convergence onto, remember, these are the axons of the ganglion cells. So there could be 6, 7, 8 rods converging onto one ganglion cells. All right. So that is not going to give you much visual acuity. Let's take a look on how that takes place. All right. Here is one ganglion cell, and here is rods. Look at 1, 2, 3, 4, 5 rods are converging onto one ganglion cell. So the visual receptive field for this one ganglion cell that's going to go back to the brain is this whole area here. Right? That's not going to give us very, right? If there's lights coming in, different light sources coming in here, we're not going to be able to get this sharp vision. Whereas for the cones and the fovea, it's usually one cone to one ganglion cell being activated. So here's, say, the receptive field for that ganglion cell and the receptive field. So light hitting here is going to activate a separate ganglion cells compared to light hitting here. Whereas in this situation over here, any light hitting here in this region is only going to activate one ganglion cell. Okay, so remember, the brain can only interpret what is getting to it. In this case, any light in this region here will only activate, send one neuron up to the brain, where light here, light here, light here will send up separate neurons to the brain. All right, and activation. Therefore, your brain can put a much sharper image and perception to light coming in here compared to light coming in to those regions where there's more rods. Okay, so it has to do with density as long as, and also with the amount of convergence. Very little convergence at the fovea, lots of convergences out on the periphery of the retina. So let's get into our neural pathways. And what we saw with our somatosensory reception is the left side of the body went to the right side of the somatosensory cortex. Almost similar in the eye, but it has to do with the visual fields. Okay, so not all of what comes into the right eye, and so this is looking almost upwards at a person. What's coming in at the right eye is not all going to the left side, and not all that's coming from the left eye is going to the right side. It has to do with what part of the visual field. Okay, right visual field, left visual field, and you can see how the light hits the different eyeballs. All right, in here, this part of the left visual field is only going to hit this part of the left eye. It's going to hit the medial side, where this part of the right or left visual field is going to hit the lateral side of the right. Right? So the medial of the left and the lateral of the right are all going to go to the left, whereas the right visual field, the medial of the right hits, and the light from the right visual field hits the lateral of the left, and those are going to go all to the right side. Okay, so that is how that is operating. Let's see, when we get into it, we can get, there are two pathways that are going. We have the tectal going here, and the geniculostriate pathway going back here. Okay, those two pathways, and they're giving us different information of what is coming in. All right, the geniculate is going to give us our what? Okay. Whereas the tectal is going to tell us where it's at, right? We got lots of visual information. Hey, we need to know where, how far away, and then we got to know what it is, right? So those are the two pathways coming in that are going to give us our different information of what we are seeing. Right, so very detailed, very complex, but we've got through it. Next, we're going to move on to the autonomic nervous system, and that will be, after that, that will be the complete of what we'll do for the nervous system. We will see the nervous system throughout the semester because we will be using it for homeostasis, helping regulate organs that keep us homeostatic, but we're not going to be specifically looking at any more portions of the nervous system after we finish the autonomic. All right.